All right. So anyway, I'm as Susan said, I'm a new PI at the University of Utah, which is right here on the picture, if you can't see it. And I've spent the last 3.5 years sort of getting my independent lab off the ground. And what I want to tell you about today are two new stories that are coming out of my lab, and they both relate to this really critical, really fascinating pathway called the Hedgehog pathway that underlies really important aspects of development and drives a number of different diseases. And the fun thing about these, working on this pathway is that I think it's turned out to involve GPCRs and kinases, which are part of many signaling pathways. But it turns out that the Hedgehog pathway uses these molecules in really unusual and really interesting ways. And so that's what I'm going to tell you about today. So let me sort of start off by taking a step back and telling you about the questions that really drive our lab as scientists. And so we're really broadly interested in this question of how animals develop. And I have to say, it's always really fascinated me, the fact that we all start off our lives as this sort of undifferentiated blob of cells. And eventually that blob of cells turns into an organism or an adult with all kinds of different tissues and organs. And so all of the cells in our bodies need to adopt the correct identity. And as you can imagine, this is a really heavily regulated process. It's really critical that this goes right, because mistakes in this process can lead to birth defects or diseases like cancer. And so really at the heart of this process is the cells need to be able to communicate with one another in embryos to be able to influence each other's fates. And one of the main ways they do that is using by sending messages to one another in the form of diffusible protein signals that activate specific signaling pathways and tell a cell what it's supposed to turn into. Should it be a skin cell, a heart cell, a liver cell, and so on. And for the last few years, what my lab has been studying is, I think, one of the most critical and really one of the most mysterious of these pathways. And that's the pathway called Hedgehog. So Hedgehog is crucial for the development of pretty much every single organ in your body. So it plays major roles in embryonic patterning, major roles in differentiation. And in adults, it also plays really important roles postnatally in terms of regulating organ homeostasis through controlling stem cell populations. And it's really critical to get the right amount of pathway activity at the right place and at the right time in order for all this beautiful biology to unfold. So if you don't have enough pathway activity during development, it can lead to birth defects. And so this here is an example of a birth defect called holoprosencephaly. This is a really severe form of the disease here. There's actually much milder forms, and it turns out to be the most common genetically inherited birth defect. And this is very frequently arising from sort of insufficient Hedgehog pathway activity during craniofacial development. And then on the other hand, if you have too much pathway activity, it often gives rise to cancer. So this is an example of a patient who is suffering from basal cell carcinoma. This is the most common cancer in North America. There's over 3 million cases of BCC diagnosed every year just in the US alone. And this cancer is driven almost exclusively by overreactivation of the Hedgehog pathway in cells in the skin. So this pathway has been studied for a long time, and through some really seminal investigation, we know now a lot about what this pathway does in development and in disease. But it might really surprise you to know this, and we actually don't have a very good idea of how this pathway works. And in fact, some of the most critical steps in this pathway, until recently, we had almost no idea how they worked at a molecular or a biochemical level. And so what my lab is focusing on is trying to understand the transduction mechanism for this pathway. And really, instead of using the sorts of genetic and organismal approaches that have been a mainstay in this field for many years, we're trying to take more of a bottom-up approach and to use biochemistry and biophysics to be able to really unravel the transduction mechanism, taking a cue from work in the GPCR and the kinase fields, for example, where those kinds of studies have really been so instrumental in understanding how those molecules work. So we want to do this because, of course, it's important to understand this pathway to be able to know how it regulates all this biology and how to control it therapeutically. But I also want to say that it's really fun to study it because it's a kind of a crazy pathway. It doesn't seem to follow any of the rules that any other signaling pathway follows. It seems to just break all the rules. And so, it's a challenge because it means that we can't just take models or ideas from other signaling pathways and simply plug them in here and have that explain what's going on. We really need to be creative and think of new approaches to be able to get at some of these fundamental questions. But I think it's also been fun for me as somebody who is interested in signaling more broadly, because it's really helped me to rethink what I thought I knew about very basic concepts in signaling, like how a ligand binds to a receptor, or how an activated receptor can signal intracellularly. And the Hedgehog has given really new ideas to think about these concepts that may apply to other signaling pathways as well. And by the way, if you want to ask questions, feel free to just interrupt throughout. It's totally fine. I actually kind of like that, so ask away. Let me summarize now some of the key steps in the pathway. This is a really oversimplified diagram, but I really want to get just the key building blocks in place as a frame of reference. So we're going to talk a lot today about SMO-Nb. So SMO-Nb is an atypical GPCR that is at the heart of regulating this pathway. It's really the key gatekeeper of the pathway activity at the cell surface. And when the pathway is in an off state, SMO-Nb is blocked by another membrane protein called PASH. And so PASH blocks SMO-Nb and prevents SMO-Nb from communicating with Gli transcription factors. These are the major effectors of the pathway that control expression of genes involved in differentiation or proliferation, things that are linked to cell identity. So that's the off state of the pathway. And then the pathway is triggered when Hedgehog ligands are made by distant cells. And so they come along, they bind to PASH on the surface of the receiving cell, and that binding inactivates PASH. And so the presence of Hedgehog is like the loss of PASH. It's a double negative. So it's a little bit tricky to think about it, but everything here is built on layers of negative interactions. So once you've lost PASH, now SMO-Nb is derepressed, it becomes activated, and it can communicate with Gli. And all of this happens in a structure in vertebrates known as the primary cilium. And a lot of you may be familiar with this structure from the work of Joe Gleason here and others, but this structure is basically like a little antenna coming out of the surface of the cell. It's a microtubule-based structure, and it's essential for Hedgehog signaling to function, and all the pathway components localize endogenously to this organelle. Okay, so within this skeleton diagram here are really two fundamental questions. The first question is, how does PASH inhibit SMO-Nb to regulate its activity? And the second fundamental question is, how does SMO-Nb communicate with Gli? So I think you can see that these are really fundamental aspects of how the pathway works. So all these actions of Hedgehog in patterning structures during development, they all have to go through both of these steps in order to ultimately lead to changes in cell identity. And in diseases, like especially in cancer, mutations, for example, in PASH or in SMO-Nb that drive some of these cancers, again, have to go through both of these steps in order to produce their effects. But for almost 30 years, we've had almost no idea, again, until very recently, about how either of these processes works. And so I'm going to, and I think it becomes clear here that this is, again, relevant to nearly all the pathways' roles in development of disease. So really crucial to figure out how both of these steps operate. So my talk today is going to be divided into two parts. The first part is going to be on the PASH SMO-Nb step. And I'm going to go through that relatively quickly because we published that actually back for some time now. And I really want to spend most of the talk on some new work understanding how SMO-Nb communicates with Gli. But it has been a lot of fun, really, as a new PI to be able to participate in having, I think, some of the first solutions to some of these longstanding mysteries in the field. So let's start with the PASH SMO-Nb connection. And so let me summarize now kind of what was known about this process. And so PASH and SMO-Nb, they're not a typical receptor or co-receptor. They don't biochemically interact. They seem to communicate through a much more unusual mechanism. And the idea in the field has been that PASH is controlling SMO-Nb by somehow restricting SMO-Nb's access to cholesterol in the membrane. And that process is somehow linked to regulating SMO-Nb's activity state. So let me go through some of the evidence for that, because it is, again, a little bit of an unusual model. So if you look at the PASH sequence, and then now, because we have structures, you can also look at the structure, it resembles these lipid transporters from bacteria that use ion gradients to pump lipids into and out of the bacterial cell. So we think PASH is doing something similar to that in animals. And it's actually been shown that PASH is able to remove cholesterol, specifically from the inner leaflet of the plasma membrane. So it seems like PASH may be doing something with transport or rearrangement of cholesterol within the membrane. The details are not totally clear, but it does seem to be operating on cholesterol in some transporter-like fashion. And cholesterol is really important for SMO-Nb because it turns out to be both necessary and sufficient to activate SMO-Nb. This is a little bit unusual for a GPCR, but cholesterol is, and I think we made an important contribution to this during my postdoc work with Phil Beachy. I was able to purify and reconstitute SMO-Nb into lipid nanodiscs and show that in that system, all you need to do is add cholesterol in, and that's enough to activate SMO-Nb to near maximal level. So again, a very unusual kind of a role for cholesterol. So we have here kind of the basics of a circuit or a regulatory interaction. We have a cholesterol transporter, and we have a molecule that's regulated by cholesterol. But there's a couple of big outstanding questions. So it wasn't really clear at the outset of this how cholesterol actually activates smoothing. So is it, for example, having physical effects on the bilayer, as cholesterol often does, and somehow smoothing is sensing that? Or is cholesterol directly binding to smoothing as a ligand, for example? And if it is binding, where is the binding site? And actually, that was really not clear because although there had been a number of structures of smoothing that were solved, and some actually had cholesterol bound to an extracellular domain, we knew that that domain was actually not absolutely required for smoothing to be activated or for PASH to regulate smoothing's activity. So the whole question of cholesterol action and binding, side binding mode was really not clear. And then also, how is it that PASH activity is affecting this process to keep smoothing off when it's supposed to be inhibited? And so the approach that we took here was, when I started my lab, was really a structural approach. And so I collaborated with Ashish Manglik, who I think many of you here in the audience know. And what we reasoned was that if we could capture a structure of smoothing in a truly active conformation, which had never been done before, that might give us some clues as to how cholesterol triggers smoothing activation. And the issue, and the key, I guess, reagent here was the development of a conformation-specific smoothing nanobody that specifically recognized the active state of smoothing. And we showed in collaboration with Ashish's lab that this nanobody actually recognizes the active state of smoothing both in vitro as well as in living cells. And then we used that nanobody to trap smoothing in an active conformation and then see what the structure looked like. When we did that, we got a pretty big surprise. We found 2 cholesterol molecules that were bound in the structure. The first was bound in the extracellular domain, and I told you about that one already. That had been seen before. But we found also a second cholesterol molecule lodged in the 7-transmembrane domain, right in the binding pocket where orthosteric agonists for GPCRs, endogenous ligands, usually are often bound. And if you zoom in on this structure, which was at 2.8 angstrom, so we can actually get a lot of detail here, you can see that cholesterol makes these contacts with a hydrophobic pocket at the bottom of the binding site. And actually, if you mutate these residues that are nearby the cholesterol, you completely wipe out smoothing activation, both in vitro as well as in living systems. So we think that the cholesterol interaction with the 7-transmembrane domain is really essential for smoothing's activation. It's the key event that's driving the activation of this molecule. And I don't have time to show this data, but it turns out that the cholesterol is also directly stabilizing a conformational change in smoothing that's very similar to the activating conformational change in other GPCRs with the outward movement of TM5 and 6, just like other, and the cholesterol is directly pushing on those helices to stabilize that change. All right, so cholesterol, again, is not a typical GPCR ligand. So it's quite hydrophobic. Many GPCR ligands are more hydrophilic. So how is cholesterol getting into this binding pocket? So it turns out that there is a tunnel, a hydrophobic tunnel that was revealed here that links the inner leaflet of the plasma membrane to the binding site that we identified in our structure. And then that was very satisfying for 2 reasons. It helped to explain, first off, how is it that sterols would be able to transit from the bilayer into the 7-transmembrane domain when any kind of transfer out of the bilayer is going to be energetically very costly and unfavorable. So because of this tunnel, it never has to do that. But also this provides, I think, a very attractive hypothesis for how PASH could regulate the occupancy of this binding site. Because remember, PASH is operating on cholesterol in the membrane, particularly in the inner leaflet. And in doing that, if PASH is able to reduce the amount of cholesterol in the inner leaflet, it's going to reduce entry into the tunnel and reduce binding to this site. So it actually, I think, makes a fairly simple and fairly sensible mechanism for how this works. So this is a pretty different role for cholesterol than the role that, so I will say, to back up for a second, cholesterol is known to regulate a lot of different GPCRs. Typically, it's thought of as it's sort of an ancillary role in sort of facilitating the action of a real endogenous ligand. But we think that what's going on with smoothing is a place where cholesterol is playing a much more central and much more direct role. And to illustrate that point, we've aligned here the structures of other GPCRs from the PDB in which there's like a bound cholesterol in yellow. And you can see from superimposing these structures, the cholesterol never occupies the same position in any structure. And it's generally around the periphery of the protein, which is also consistent with this ancillary role that I talked about. However, if you look at smoothing, we have 2 cholesterol molecules bound in specific locations. And the new one that we identified in the 7-transmembrane domain corresponds really nicely with the endogenous agonist from other GPCR structures that were crystallized in their active states. And you can see that from the alignment here. So we think this is a situation where cholesterol is not sort of this passive glue that's lubricating the membrane, but it's really more like the key activating ligand for smoothen. So it's able to really directly activate a GPCR. And this is, I think, a new idea that we're really interested to explore throughout the receptor superfamily to see if cholesterol might regulate other GPCRs or other membrane proteins in this manner. So I want to finish this part of the talk by kind of talking a little bit about therapeutics. And this has been another really exciting aspect of this structure because it led to some real, I think, hypotheses for how we could develop better drugs to treat hedgehog-dependent cancers in the clinic. So smoothen is a major oncology drug target, and there's a number of FDA-approved small molecules that directly bind to smoothen and shut down its activity. And our structure revealed how these molecules likely work, because if you superimpose the structures of smoothen bound to these inhibitors with our structure of smoothen bound to cholesterol, you can see that the inhibitors bind largely in the upper part of the binding pocket, but they all extend a functional group downward that clashes with the cholesterol in the structure. So the way that they work, and this was not known before the structure, is that they're simply competitive antagonists for the endogenous sterol ligand. So another problem with a problem with these inhibitors is that they all tend to work in the short term, but eventually patients develop resistance to them. It's very easy to do that. You can sort of see why that's the case in the structure, because the overlap between the endogenous ligand and the inhibitor is very minor. So it's very easy to mutate, for example, the upper part of the binding pocket and wipe out inhibitor binding while sparing the cholesterol interactions deep in the pocket that are needed for function. So what we think is that if we could figure out a way to drug the deep part of the pocket and smoothen, that might be a much better way to avoid resistance, because in that case, it would be a lot harder to uncouple the binding of the endogenous ligand from the binding of the inhibitor through a mutation. And so fortuitously, it turns out that there's already an inhibitor around that acts like that. And that inhibitor is called SANT1. And you can see the structure of SANT1 here superimposed with our structure on the right. And you can see that SANT1 binds really deep in the pocket in a manner that very nicely overlaps with the cholesterol, much more extensively than the existing inhibitors. And so we're now hypothesizing that SANT is going to be better at avoiding resistance in the clinic compared to the existing standard of care. And that's a hypothesis that we're testing now using a number of cultured cell and in vivo models of hedgehog-dependent cancers. And so the thing I like about this is that this is an insight that I think really came out of just the understanding of how this protein actually is activated in the first place. And without that understanding, SANT just looks like a curiosity. But now I think we have the idea that it's something that could be very, very useful. And we've taken the first steps towards this by now showing that all the existing therapy-resistant smoothen mutations, isolated in the clinic from patients, are now sensitive to treatment with SANT1. And so in this assay, we've now taken smoothen knockout fibroblasts, and then we transfect in wild type or mutant forms of smoothen, along with a GLI transcriptional luciferase reporter. So in this assay, when you put in wild type smoothen, you can see you get a nice activation of the reporter in green. It's blocked by vismodegib. That's one of the existing inhibitors in magenta. And it's also blocked by SANT1, the deep pocket inhibitor, in cyan. So now we put in all of these therapy-resistant mutants, and you can see in all cases, the vismodegib inhibition in magenta is not as good as the SANT inhibition in cyan. And so SANT is actually able to block these existing resistant mutations in the clinic. So we think that's an encouraging first step. And so we're now keen to really test SANT in vivo and in cultured cells in tumor models. And the real test here is going to be whether or not with long-term treatment with SANT1, whether or not we're going to be able to forestall or avoid resistance more effectively than we could with the existing standard of care. So those are experiments that are in progress right now. Okay, so I'm going to move on. Oh, yes?
Just a quick question. As I recall, Hedgehog is also cholesterol modified.
That's right.
That dependent cholesterol play any role in the regulation?
It does. Yes and no. So you're right. So Hedgehog is cholesterol modified. Actually, Hedgehog itself has two lipid modifications. It has a cholesterol on the C terminus, and it has a palmitate on the N terminus. So the cholesterol is actually dispensable for signaling. But what it is important for is regulating the release and the distribution of Hedgehog proteins throughout tissues. The palmitate, on the other hand, is essential for signaling, and that actually interacts directly with the part of patch that's involved in lipid transport. So, yeah. Good.
We have a question almost. So I will try and I'll move forward for a second. Okay, Paul.
Oh, sorry, I was muted. A very simple question. To what extent do you think what you've just described for smoothing is unique to smoothing, or is there are there other GPCRs that may share that same site or something akin to that for cholesterol?
Okay, yeah. So Paul was asking if this is unique to smoothend or if other GPCRs might show this. So we don't have the answer to that. I think it's really difficult to know that from looking at sequences because there have been motifs that have been described for cholesterol binding and cholesterol recognition, but smoothend actually doesn't seem to correspond to that. And I think actually, to be honest, I think a lot of those motifs have a fairly limited predictive value. You really need to be able to show biochemically or structurally that these things bind in order to be able to get at that. So I think actually the thing that's going to help this move forward is probably more structural information and more biochemistry to get at other GPCRs that might bind cholesterol.
Thanks.
Right. So these are mouse embryonic fibroblasts, and so they're just from smoothin knockout mice, and so they don't have any smoothin, and so they just rely completely on exogenous smoothin to be able to signal. But we are trying to extend these studies to cellular and eventually organismal models of cancer to be able to test whether or not this holds true in a real cancer cell context. Yeah. All right. If there's no more questions, I will move on to the next part now. So this is the new work from my lab talking about how it is that smoothin communicates with the other really big mystery in this pathway. And this turns out to involve a molecule that many of you are familiar with, protein kinase A or PKA, but in again, a really unusual way. And we published actually the first part of this in PLOS Biology a little bit in April of this year. So this was coming from my lab. And then the second part is also this preprint. This is a result of our collaboration with Susan's lab. It has been really amazing. And that we just put online on bioRxiv in the beginning of July. And I'm going to kind of go back and forth between these papers because you kind of need both of them to be able to get a full picture of what's going on. All right, so let me talk to you first about PKA. So PKA has been appreciated for a long time as the critical negative regulator of GLI in the Hedgehog pathway. So the way this works is that PKA phosphorylates conserved sites on GLI and stimulates the processing of GLI into an inactive repressor form that's unable to activate transcription. And this event happens in the primary cilium. This is a really nice paper from Bao and Wang's lab, data from a paper showing that if you make a phosphospecific GLI antibody that recognizes these PKA sites on GLI, you can see it light up the primary cilium in the pathway off state, and then the signal is actually reduced when you turn the pathway on. So it's in the cilium where PKA phosphorylates and inactivates GLI. And this is actually reflected also at the genetic and at the phenotypic level. If you make mice that are lacking both the C alpha and the C beta form of the PKA catalytic subunit, or the C subunit as I'll refer to it, these mice die very early in development of massive overactivation of the Hedgehog pathway. And this phenotype is actually just as strong, if not stronger, than the phenotype that you would get from just knocking out PASH. So this is something I have to say has always kind of amazed me, because I think of PKA as this very multifunctional enzyme that sort of is participating. It's got its fingers in every signaling pathway that you can think of. But what the genetics is telling us here is that the key role of this protein in early development is actually to restrain the Hedgehog pathway. So I always have found that kind of amazing. Anyway, so you have a PKA here, and you have smoothend. And so one early idea in the field was that smoothend might activate GLI simply by turning PKA off. And that's a very appealing idea, right? Because smoothend is a GPCR. It's in the same superfamily. And GPCRs are very good at inhibiting PKA through the use of inhibitory G proteins like G alpha I, O, and Z. But the problem with this model has been dogging the field for many years, is that we know that if you inactivate these inhibitory G proteins, either through genetics or pharmacology, you don't prevent smoothend from activating GLI. And so that's really left the whole field kind of wondering what's going on here. I mean, is smoothend even communicating with PKA or not? Is smoothend maybe activating GLI through some other independent pathway? So what's going on here? So our hypothesis was that maybe smoothend has a way to inhibit PKA that just doesn't depend on G proteins. And so that was the basis of our plastic biology paper. We investigated this hypothesis by trying to focus on whatever was going on immediately downstream of smoothing. We figured that that was going to be the key to understanding or unraveling this mystery. And so this is all going to be reviewed, so I'll go through this pretty quickly. But how is it that GPCRs typically block PKA? So again, a GPCR would couple to an inhibitory G protein, and that would inhibit adenylate cyclase and then lower levels of cyclic AMP. And that will promote the formation of inactive PKA holoenzymes that are made up of the regulatory or the R subunit and the catalytic or the C subunit. Okay, so we all know that, but then our hypothesis now is, does smoothen have a way to block the C subunit of PKA without using this pathway? And so to address to test this idea or to ask this question, we attempted to completely bypass the G protein dependent signals and see if smoothen still has a way to inhibit the C subunit. So we're going to basically take all this out of the equation, and then we'll be left with something like this. So to test this idea, we needed a way to measure PKA activity, and we used for that a conventional Krebs luciferase reporter assay, where the active C subunit phosphorylates Krebs, and that turns on luciferase. Okay, so first things first. So we showed now in 293 cells that if we overexpress the C subunit, we get nice activation of the Krebs reporter. So that makes sense. And then now we're going to test whether or not we've bypassed the G protein dependent signaling by using a different GPCR, the M2 muscarinic receptor, which is a classic GI coupled GPCR. So if we transfect the muscarinic receptor into these cells that are overexpressing the C subunit, we fail to see inhibition of the reporter, either in the basal state or in the presence of the ligand carbachol, shown in gray there. So that shows us that we've effectively bypassed the G protein dependent signals here. And again, the idea here is that by overexpressing the C subunit, we're exceeding levels of R subunit in these cells and exceeding their capacity for regulation. So we just have this positively active C subunit. All right, so now what happens with smoothen? So we're going to put smoothen in. And one thing I want to point out here is that these 293 cells, they have very low levels of endogenous PASH, and we're overexpressing smoothen. So in this system, smoothen is constitutively active. It's essentially bypassed the PASH that's present. What we found was when we did this, we very dramatically blocked the activity of this Krebs reporter with smoothened expression here. And that effect is specific because we can block it by adding a specific smoothen inverse agonist called catcyclopamine, which competes with a cholesterol, as shown in red here. So this is what gave us the first indication that there's something going on here where smoothen has a way to regulate the C subunit in some unusual fashion. And I'll also point out here that we did the same experiment in 293 cells completely lacking all 13 G alpha proteins, and we got exactly the same results. So that really confirmed this idea that there's G protein independent signaling. So we next tested the idea, okay, so how does the signaling work? So we tested the idea that maybe smoothen directly interacts with the C subunit. And to do that, we used Brett assays with nanoluciferase tagged smoothened, and then YFP tagged versions of the C subunit or various other proteins. And in these experiments, as a positive control, we used our nanobody because we know that binds really nicely to the intracellular face of the transmembrane domain. And our negative control here was beta-arrestin 1, which is a protein that we can express at very high levels in these cells, but really does not appear to interact with smoothen. So this is what that experiment looks like. So as expected, the negative control beta-arrestin in black shows just background levels of Brett. The nanobody in gray shows nice Brett with smoothened. And then the C subunit of PKA actually showed such a strong Brett signal on this experiment that it even exceeded the positive control. So that was a really great day in the lab when we got this result. We went on to then map the structural determinants of this interaction. It turns out to all be in smoothen's intracellular cytoplasmic domain. And in particular, there's a domain that we call the proximal cytoplasmic tail, or the PCT. And this is really the primary binding site for the C subunit in this Brett assay. And this is also the region of the smoothen tail that is essential for communication with glee. So there's a lot of examples of GPCRs that bind to PKA, but bind to the holoenzyme through R subunits and through ACAPs, for example. So we were thinking maybe that's what's going on here. Maybe the C subunit is just being dragged along with a more conventional kind of a GPCR holoenzyme interaction. However, that's not the case, because if we do Brett between smoothen and the R subunit shown in gray here in the saturation experiment, we really don't get any Brett signal, whereas the C subunit in black gives a very nice signal. So smoothen has a way to directly interact with the C subunit. Or that, sorry, not directly interact, that's coming later, but it has a way to Brett with the C subunit. And we confirmed these experiments using confocal microscopy and doing colocalization studies. So in these studies, if we overexpress the C subunit in 293 cells, it has a fairly uniform cytoplasmic distribution. But if we co-express smoothened in these cells, smoothened is at the membrane in magenta, you can see that the C subunit gets dragged from the cytoplasm to the membrane in these experiments and co-localizes really nicely with smoothened there. And that effect depends, again, on the smoothened intracellular domain that we know is critical in the bread assay and critical for signaling to GLI. So these experiments show colocalization in 293 cells at the plasma membrane. We've also, oh, that's odd. Oh, there we go. Good. We also have been extending these studies now to ciliated cells to look more in the endogenous location for the pathway to be active. And in these IMCD3 cells, which are a ciliated kidney cell line, we also see really nice colocalization between smoothened and the C subunit in the cilia. So I think that makes us feel more confident that what we're seeing is something relevant to the physiological pathway as well. Okay, so what are the consequences of membrane localization? So two things. The first thing is that we think that so GLI is a soluble protein, smoothened is a membrane protein, and by dragging the C subunit to the membrane, smoothened is essentially going to be restricting the C subunit's access to GLI and thereby preventing or hindering GLI phosphorylation. So that idea of sequestration was the idea that was the central idea in our PLOS biology paper. But it turns out that that's only really a part of the story. And so this is what came out of our collaboration with Susan's lab, but it turns out that smoothened has a way to directly inhibit the activity of the C subunit that it binds to. So before I tell you how that works, let me remind everybody here that typically in cells, inhibitors of the C subunit are pseudosubstrates. And so they bind in the active site of the enzyme like a substrate, but they just take up space, they interrupt the catalytic cycle of the enzyme, and they prevent further rounds of catalysis. And the famous examples of that are the regulatory or the R subunits, as well as the PKA inhibitor proteins or the PKIs. And the regulation is different. So the R subunit's regulated by cyclic AMP, PKI is more or less constitutive, but both of these proteins engage the active site through a very similar structural mechanism. And that was highlighted here in Susan's really important 1991 structure of the C subunit bound to PKI, where you can see that it really engages the active site just like a substrate, in a similar manner to how a substrate would. So as Susan alluded to, in the early stages of the pandemic, we had an early version of this story about PKA, and I wanted to talk to an expert in the field and see what they thought about it, because PKA has been studied for so long, and we're really kind of newcomers to this field. And so I showed Susan our data, and she in particular asked to see the sequence of the domain of smoothened that we thought the binding site was located to. So we showed her this sequence alignment here. And within about 15 seconds, she had zeroed in on this motif in here that bears a very, very striking resemblance to PKI. And I have to say that this just totally took me by surprise. And I vividly remember this moment where really just everything kind of clicked into place. And it's really been an amazing insight to be able to make. So let me zoom in now so you can see this in more detail. So now we have an alignment here with smoothened on top and PKI alpha on the bottom. There's a number of key similarities between them. So first off, both smoothened and PKI have an alanine, a non-phosphorylatable residue at the P site in this motif. In addition, at the P+1 site, you have a hydrophobic residue in both smoothened and PKI. And then at the -2 and -3 positions, you have arginines. And all these residues are critical for high affinity binding, in addition to a bulky aromatic residue at P-13. And in addition, this region of smoothened is predicted to be strongly alpha helical. And the same region of PKI is alpha helical, and that alpha helicity is essential for high affinity binding. So, even though to me, really at the start of this looked like just a string of letters, it was kind of amazing to see that somebody was sort of looking at things in the right way could be able to come up with this analogy. But it really makes a lot of sense. So we immediately hypothesized here that smoothened is going to stick this PKI motif, as we call it now, directly into the active site of the enzyme and shut down catalysis. And that makes perfect sense because we also knew from other work in the field that if you truncate smoothened from the C terminus inward, and you ask how far can you go before smoothened cannot communicate with GLI anymore, it turns out the farthest you can go is right here to that leucine. And if you go any farther than that, you no longer can communicate with GLI. So of course that makes a perfect sense now because you're going to be interrupting what we're now calling the PKI motif. So immediately, Susan and I started collaborating. We pulled a number of other labs in here to test this idea as well. And it's been so much fun to be able to be part of this. And so one thing that we started to do right away was to conduct some in vitro studies to test whether or not smoothened actually binds to PKA as a pseudosubstrate. And it does. So this is an SPR experiment done by Daniela Bertinetti and Fritz Herberg's lab, showing that a wild type version of the recombinant smoothened cytoplasmic tail binds really nicely to the C subunit. And that binding is definitely a pseudosubstrate form of binding because it's blocked, or it completely depends on magnesium and ATP, which I'm not showing here. And it's also blocked by mutation of the key PKI residues in smoothen, namely the arginines at -2 and -3, and the tryptophan at -13. So we call this the WRR mutation. You can see here that that really wipes out the signal in the SPR assay. And I don't have time to show the data here, but we've done very similar experiments with in vitro activity assays of PKA enzymatic activity. And we also see that the smoothened tail can inhibit PKA enzymatic activity in those assays. So one thing that, and those are the residues. So one thing that surprised us here at first is that the KD for this interaction is rather weak. It's around 800 nanomolar, which compared to a typical PKI is more like 1 nanomolar. So we were wondering what was going on here. And it puzzled us at first, but eventually what we came to realize is that this low affinity is likely really critical for ensuring that this interaction does not happen spontaneously in solution, and for creating a situation where you only get an interaction when smoothened is in an active state. And I'll get more into that in a few slides. So in the meantime, while Susan and Fritz and John Luigi Veglia were doing some of those experiments, in my lab, we started to look at whether or not the PKI motif was important for interacting with PKA and signaling in living systems. And this is now a Brett assay looking at either the wild type or the WRR mutant form of smoothened in a Brett assay. And you can see that the WRR mutation almost completely wipes out interaction with the C subunit, shown in tan. Critically, interaction with nanobody 2 is more or less normal, and that is minimally effective. And that's a really important control. It shows that this mutant form of smoothened is able to be expressed and trafficked properly, and also can undergo an activating conformational change, because this is, again, a conformation-specific nanobody. So that's, I think, a really good internal control to show that the mutation is not simply messing things up in a non-specific way. And we got similar results. Odd. Here we go. Okay. We got similar results in the colocalization assay, showing that the WRR mutation prevents colocalization between smoothened and the C subunit at the plasma membrane, and that's quantified at the right. Okay, so what is the significance of this PKI motif interaction for Hedgehog signaling in vivo? So we haven't looked at that yet. So to address that question, we made use of a zebrafish embryological assay. So this is now looking at zebrafish muscle development, which is very, very explicitly sensitive to Hedgehog pathway activity. And so if you look in wild-type zebrafish embryos, they develop muscles normally, marked by PROX1 in magenta and engrailed in green. And as expected, if you knock out smoothened, you prevent you prevent this process from happening, and these cells don't develop. So this now gives us a background to test mutant smoothened. So first, if we put wild-type smoothened into these knockout embryos, we can rescue the patterning. But if we put the WRR mutation into these WRR mutant into these embryos, we fail to rescue the patterning defects. And I'll say that we got similar results in a cultured cell as a glial transcriptional reporter assay as well. An additional mutation that we made here is we actually also mutated the P site alanine back into a serine. And so that's something that is going to reduce affinity, just make it get phosphorylated and fall off. And that also has a very, very similar phenotype in all of these assays. So I really like that experiment because, again, it sort of shows that you need this sort of deeper understanding of PKA and how PKA works in order to be able to design and interpret those kind of experiments. So it really confirms for us this idea of a pseudosubstrate interaction. All right, so a big question now is, so we've demonstrated this interaction, but how is it linked to smoothened's activity state? You can't have this interacting and inhibiting PKA all the time, because if you did, then the pathway would be on all the time, and you would get ectopic Hedgehog pathway activation. So it has to be tied to smoothened's activity state. So that puzzled us for a little while, but it turns out the answer to this is a molecule that many of you are familiar with called GPCR kinases, or GRKs. So GRKs are famous throughout the GPCR superfamily for phosphorylating GPCRs selectively in their active state, and usually triggering desensitization of these receptors by recruiting accessory proteins like beta-arrestins that bind specifically to the phosphorylated form of the receptor. So in the case of smoothened, we knew that GRKs 2 and 3 were essential for smoothened Glee communication. There was some really clear genetics and pharmacology to indicate that. But beta-arrestins didn't seem to be involved in this process. And so we hypothesized that maybe GRK phosphorylation, instead of triggering beta-arrestin recruitment, is going to be triggering recruitment of the C subunit. And that would potentially explain all of this. And it turns out that that appears to be the case if we go back to that Krebs reporter assay and we test the effect of a specific GRK inhibitor, GRK23 inhibitor, compound 101 in purple, we strongly reverse smoothen's ability to inhibit this reporter just about as well, or perhaps even better, than using a smoothen inverse agonist. Also, compound 101 blocked the co-localization of smoothened and the C subunit of the plasma membrane, and it also reduced the Brett signal as well in data that I don't have time to show you. So this I think ended up being pretty interesting because a lot of people think of GRKs really only in terms of facilitating arrest and recruitment. But I think what's going on here is a little bit of a twist on that theme, where GRK phosphorylation is triggering a different kind of a protein-protein interaction, but a positive interaction that's really with another molecule, but undergoing the same kind of regulation. I will also say that we mapped the critical GRK23 phosphorylation sites on smoothen by mass spec, and we found 7 of them shown in orange there. You can see that they're all outside of the PKI motif itself, so they're not directly influencing the pseudosubstrate interaction. What we think that the GRK phosphorylation is doing is sort of facilitating this interaction by essentially helping to recruit the C subunit to the plasma membrane, and now turning this from sort of a three-dimensional system, three-dimensional diffusion system, to something that's much more 2D, where these interactions are going to be a lot more confined. And now the PKI motif has a much higher chance of binding effectively. And so essentially what's going on here is that you have a couple of low affinity interactions. The PKI interaction is low affinity, perhaps the GRK phosphorylation induced interaction is low affinity, but by compounding them together, you now create a much more favorable situation to interact. And furthermore, because GRK phosphorylation is linked to smoothen's activity state, this provides a really nice activity-dependent switch so that this interaction only happens when smoothen is in an active state. So to take a step back for a second, what I've told you about in the second part of the talk is a mechanism where a phosphorylated receptor tail is sequestering and inhibiting a kinase. So when smoothen becomes active, when it binds to its sterol ligand, it's recognized by GRKs 2 and 3, that triggers phosphorylation of the smoothen PCT domain, and that helps to recruit the C subunit to the membrane. We think this is happening in the primary cilium. So to recruit to the ciliary membrane, where now this low affinity PKI motif can now bind much more favorably due to the increased crowding. And that triggers inhibition of PKA and then finally dephosphorylation of Gli and Gli activation. So again, smoothen has come up with a way to regulate PKA activity without needing to use G proteins. So it's a really unusual and in many ways unprecedented mechanism in the GPCR superfamily. But it turns out it's not a unique mechanism. And a very similar process is at play in another critical developmental and stem cell and cancer pathway known as the Wnt pathway. And many of you know about Wnt already from Carl Willard's work here at UCSD. But it turns out that during Wnt signaling, Wnt binds to a receptor complex made up of frizzled and LRP5 and 6. And it turns out that the LRP5-6 cytoplasmic domain is also a pseudosubstrate inhibitor of a kinase. And that kinase is GSK3 beta. And by inhibiting GSK3 beta, you prevent the phosphorylation-induced inactivation or degradation of beta-catenin. And it turns out that this interaction is also quite low affinity in solution. It's also micromolar affinity, but it's strongly enhanced by phosphorylation of the LRP tail, as well as recruitment of GSK3 beta to the membrane through other protein complexes that are not indicated here. So it's pretty amazing that actually the same or very similar regulatory principle appears to be underlying both pathways. And this is something that really took me by surprise, because I didn't really think of Hedgehog and Wnt as signaling intracellularly with a similar mechanism. And the molecules involved are completely distinct from one another. And the cell biology is also very distinct because Hedgehog uses the primary cilium, whereas Wnt uses the plasma membrane and also multivesicular bodies. But it turns out that those are, I think, in many ways, sort of surface level differences. And when you get down to the deep roots of these pathways, they really do seem to share a very similar regulatory principle mediating their intracellular signaling. And that's really ended up being a surprise and something that's making us think a lot more about why that's the case. So why is that the case? So we don't know. But one idea, okay, so why utilize a mechanism like this? So our leading idea now is that this is really all about ensuring signaling specificity and reducing unwanted crosstalk. So both of these pathways are using really pleiotropic kinases that are involved in many signaling pathways. And the challenge is, how do you regulate the kinase without necessarily affecting all these other processes that involve PKA or GSK3 beta? So one way to do that is by only restricting inhibition to this small pool of kinase that is directly bound to the receptor. And by doing that, you're going to essentially insulate the inhibition of each of these kinases from all the other pools of kinase throughout the cell. And that's going to be a situation that's desirable, because you could imagine if smooth activation, for example, inhibited too large of a pool of PKA in the cell, you're going to now affect every PKA-dependent process that you can think of. And that's not so good. So in the end, we think that this actually makes makes sense. At least we can rationalize it from this perspective. All right, so to kind of return to some of the questions I asked at the beginning. So we were talking about how is it that PASH regulates smoothing, and how is it that smoothing signals intracellularly? And we've actually come, I think, a pretty long way in the last couple of years in answering these, in starting to answer these questions. I've shown you today that PASH regulates smoothing's access to cholesterol in the membrane through the regulation of binding to a novel site that we identified in the 7-transmembrane domain. And I've shown you that smoothing signals to gli, intracellularly to gli, by regulating PKA in this very unusual fashion that involves a dual sequestration and inhibition mechanism centered on physical blocking of the PKA active site. So this has led to now a number of future directions in my lab that we're pursuing. So on the PASH smoothing project, we really don't understand nearly enough about how PASH works and how is it that PASH alters smoothing's access to cholesterol. This is a really important key question for the field to answer. And one that's going to require, I think, some real deep dive into the transport mechanism to try to understand how it works. And I told you a little bit about our efforts to try to use these smoothing deep pocket inhibitors to block oncogenic signaling while avoiding resistance using some cancer cell and in vivo models. And then on the PKA project, we really want to understand the structural basis for how these complexes form and how they're regulated. And not just because I think a structure is nice for its own sake, but because this is the first example of a GPCR or any membrane receptor that directly binds to and inhibits PKA, we have no models to draw on. And so we have no idea, for example, what the critical domains are for interaction besides the PKI motif itself, why the interaction is so low affinity, and how it is that GRK phosphorylation triggers what we think is going to be a higher affinity binding state. So structures are going to help us to get at those questions. And we also want to understand more about how smoothing and the C subunit interact in the native environment of the primary cilia. We've taken some steps towards that, but we're starting to use now some advanced microscopy techniques to be able to understand the dynamics of those interactions in cilia in more detail. And then finally, is smoothing the only example of a GPCR that does this? So we don't know, but my suspicion is not, because if you think about it, there's hundreds of GPCRs that communicate with PKA. And it's really been assumed in the GPCR field that this occurs exclusively through the action of G proteins. But I think what we've shown here is that GPCRs can actually regulate PKA localization and PKA enzymatic activity through other mechanisms. And it's possible that other GPCRs are doing this too, either apart from or maybe in addition to signaling through G proteins. And this could be a way to sort of impose a level of spatial regulation on PKA signaling that would be sort of somewhat, that would sort of add to the cyclic AMP signal and the cyclic AMP regulation that already exists. And that could help to bias phosphorylation toward or away from specific regions in the cell. That could mean different things to the cell than just phosphorylation everywhere. So it could also be a way to sort of allow GPCRs and PKA to mediate and couple to new kinds of biological outputs. And so this is, I think, going to keep us pretty occupied for some time. But I think one last point I want to end on here is that I thought, when we began these studies, I thought that I understood at least the basics for how GPCRs are activated by ligands and how GPCRs can signal intracellularly. And I think one of the real privileges of working on smoothing has been that realizing that I don't understand how these processes work at all. And there's actually a lot more to learn about these concepts than what we take for granted, when we read textbooks and review articles that emphasize sort of the canonical paradigms. And there's really still a lot of room for surprises in understanding both of these processes. So I just want to end now by thanking the people that did the work in my lab. And so, and I want to also thank our key collaborator, Ashish Mangwit at UCSF. And so Ishan Deshpande from his lab and Danielle Hadin from my lab were key players in the Smooth and Structure project. And then, of course, I really want to thank Susan and Jessica in Susan's lab, as well as Fritz Herberg and Daniela in his lab, John Luigi, and also in our lab, Corbin Arbiseth, John Happ, and Isaac Nelson were really the people that led the PKA projects that I told you about. I also want to thank our funding sources, in particular a MIRA grant from NIH and a new R03 that we got very recently to explore some of these GPCR kinase interactions. And lastly, I just want to say that we are a growing lab. We're recruiting postdocs. And so if you or anyone that you know is interested in some of what I talked about today, please feel free to get in touch, and I would love to hear from you. All right, thanks a lot.
We'll discuss it more. So the GPR 161
Yes.
would be another example, but that would be the other way around.
Right. So just to clue everyone in here. So what Sylvia was referring to is there's a constitutively active GPCR in the Hedgehog pathway called GPR 161. And so that couples to G alpha S. And that GPCR actually leaves primary cilia. So it's in the cilium. And it actually leaves primary cilia when the pathway is activated through a mechanism that's not entirely clear yet. So we think what that's going to do is that's going to help to lower levels of cyclic AMP in the cilium when the pathway is in the off state. And that's going to help to favor or kind of reduce the amount of free C subunit to make it easier for smoothened to bind to the C subunit. So, right, it has an A cap. And so the A cap binds to the R subunit. And I think, I mean, I don't exactly know how to think about that yet, but currently what we're thinking is that the exit of GPCR 161 from cilia is going to block PKA activity by reducing cyclic AMP, but it's also going to continue to drag C subunit out of the cilium with it. So I think these are all mechanisms that kind of help to amplify the signal from the PKI motif. So we think that the PKI motif is really the central or the fundamental part of the mechanism. It's the part that's essential in all the systems that we've tested, but then GPR 161, that kind of, it provides an additional level of regulation that could help to tune this interaction, in different contexts. Sorry? Right, yeah. So, right, yeah. That's what we're thinking, is that basically by recruiting the holoenzyme and then dragging it out of the cilium, that it's going to help to kind of basically create a situation where the C subunit activity is as low as possible. Yeah.
But the GPR 161 seems like 3R. It doesn't seem to have C with it, so it's like C3R with cyclic AMP bound to it.
Oh, I thought it also bound to C.
No,
It doesn't?
It's mostly bound to the R. It's like Gene Zhang's. Maybe that's a little way to cyclic AMP is all around this.
Yeah, so that's another idea too, is that possibly a cyclic AMP microdomain or nanodomain organized by R might be sort of broken up or re-regulated upon GPR 161 leaving the cilium. That's another idea for sure. Yeah. Oh. Go ahead.
Oh, sorry. Yes, I didn't hear. Yeah, so my question was about where GLI is phosphorylated by PKA. In your model, it would have to be only phosphorylated in the primary cilium. Otherwise, once you're not inhibiting PKA.
Right. Okay. I couldn't quite hear you. I think what you're asking is where in the cell is PKA phosphorylating GLI, and is that only happening in the cilium versus is it happening throughout the cell? Is that
right? Exactly.
Right? Okay. Yeah. So you can force GLI to be phosphorylated by PKA outside the cilium or even in vitro with PKA. But endogenously, yes, it's only happening in the primary cilium, or at least that seems to be the pool that's critical for regulating GLI activity. So what it is that's actually triggering PKA to be active in the cilium and to phosphorylate GLI in that compartment is actually not clear. And that's, I think, a really important question to try to figure out in the future, is what is creating this environment in the cilium where you have this free C subunit that can phosphorylate GLI? Is it, for example, a cyclic AMP domain in that organelle, or is it possibly something else? So that's a really important question to answer going forward.
But are the R subunits localized to the primary cilium as well?
They are, yes. So you can find R subunits in primary cilia. The real question, I think, is what the cyclic AMP concentration is in cilia. And there's actually been a couple of different attempts to look at that. They have gotten somewhat conflicting results. So it's not clear right now if the cilium has really got a higher level of cyclic A compared to the rest of the cell, or if it's more similar. I also want to point out those kinds of measurements are pretty challenging to do, and they don't take into account the possibility of microdomains that we know exist elsewhere in the cell. And I think it's going to require more advanced imaging tools to be able to really get at that.
Great. I had another question about the GRK phosphorylation sites. I mean, looking at Susan's structure, it looks like those phosphor residues would come into direct contact with the C subunit as it trails across.
Yeah. So
the question is,
back to the let me see if I can go back here. So we actually don't have a structure yet of this complex. I mean, we have a structure from 1991 of just the peptide bound to let's stop working. So we have the structure of just the peptide, but we really have no idea where those other sites are. And in fact, I guess another point to make here is that other than that helix, there's really not, well, there's not any.
Sorry. I was trying
I
to
got-
turn him up, but not you.
I got biblically loud there for a second. Other than the PKI helix, there's actually no other similarity at the primary sequence level between PKI and the smoothened tail. And so we actually don't really know where the GRK phosphorylation sites are in 3D at all. So that's part of the reason why we think it's so important to get a structure, because we won't know otherwise.
But did you measure the affinity of the triphosphorylated form for PKA?
We have not yet. So that's a really important experiment. We want to do that. But one of the issues here is that unlike other kinases, GRK phosphorylation is really inefficient on peptides in solution. You really need to have a membrane, especially membrane lipids, and you need to have a GPCR in an active state, because it turns out that the GRK directly docks onto the active state of the GPCR to be phosphorylated. And so those kind of measurements are difficult to do in vitro, but we're trying to do them now with purifying and reconstituting full-length smoothened into nanodiscs and doing GRK phosphorylation that way. So we definitely want to do that. We definitely want to get a KD to be able to get at that. Also, another important thing in that regard is that most of our measurements up to this point have used PKA produced recombinantly in bacteria that lacks the N-terminal myristoylation. And that myristoylation may be very, very important for triggering or facilitating membrane recruitment. So that's another idea that we want to try to pursue as well in terms of thinking about that.
That's a good idea. Yeah, that's great. Thanks very much.
Thank you.
So you have three lipids. You have the palmitic acid that's on the 8th helix.
Yep.
You have cholesterol, and you have the receptors there. So to me, the structure is going to be interesting how those lipids.
Absolutely. And where they are in the membrane relative to each other. Absolutely. Yeah.
And it's relevant for Tony's question about, if you go back very early, one of the early PKA experiments with the resting oocyte, the frog oocyte.
Yeah.
It's like the same kind of situation. You have PKA active, you just inject PKI into that and the cell types divide it. I mean, it's a very similar thing. You have a constitutively active, somehow constitutively active system.
Right. I think that's been something interesting to realize here is in doing this work, I realized that now we understand more about the on state of the pathway, but the off state of the pathway is actually not nearly as clear as we thought it was. And there's definitely going to be a lot to figure out there. Yeah. Irina. Right. Yeah. That's a really good question. Right. Right. Yeah, so we think, so it's a little bit hard to say. We think probably not. And the reason is that, if you do experiments to inactive, so if GI signaling was a really, really central part of this process, you would expect that inhibiting GI signaling through things like pertussis toxin or pertussis toxin plus G-alpha-Z knockout would block the pathway. And that experiment has been done many times by many labs, and in some cases they can see small effects, but they never really completely wipe out the signaling, and in many cases they see no effect at all. So I don't necessarily want to say that it doesn't have any effect, because I could imagine a situation where, for example, smoothen could couple to GI proteins, and maybe that helps to again lower the amount of free C subunit in the cilium to facilitate the action of the PKI motif. But I think that's a critical sort of distinction to make here, is that all the efforts that people have made to inactivate G proteins haven't produced these enormous effects. They may produce subtle effects, but the PKI motif interaction here in the pseudosubstrate part of this, we think, is probably the fundamental part here. And this may be, for example, another layer of regulation on top, the same way that GPR161 is an extra layer of regulation on top. That's right, because we can see that, like for example, in cells that lack G proteins, we can still see effects of of compound 101, for example. So, yeah. Yes.
Can I say something? Paul said they can't hear on Zoom, so can you just speak up louder?
Oh, and I can also repeat. I'll repeat the question. Yeah, no problem. Yeah. Absolutely. So the question was whether or not we think it's important to include the lipid bilayer in our thinking about the ways in which the smoothing interacts with PKA and with the cholesterol. Is that right? Yeah, absolutely. And you know, a lot of this has sort of ignored, or not ignored, but sort of, you know, minimized, I guess, the sort of cell biology contribution of different, you know, microdomains of lipid domains in the membrane, things like that. But absolutely, I think that's going to be a critical part of this. You know, in our bottom-up approach, I think we're not there yet, but it's definitely a place that we want to go. And so I alluded to trying to do some more experiments with purifying and reconstituting smoothened and nanodiscs and looking at the interaction with PKA, for example, in that system. I think those kinds of things are going to be really crucial to kind of take this, you know, sort of simplified model and then, you know, translate it into something that is more, you know, more reflective of what's going on in a real cellular situation. Yeah.
So another thing, I hadn't really thought about, but usually the free-C subunit is degraded by E3 ligases.
Yes. Yes.
There are two sites of that point. So why doesn't it get degraded?
Yeah, that's a good question. I mean, that would be a very interesting hypothesis to test. And actually, we're gearing up to test some of those events as well. It seems that from work from other labs in the Drosophila system, it actually seems that binding to smoothened actually stabilizes PKA. And so it seems to almost be the opposite. I don't know why that is. It's an interesting question, but it does make me think a little bit more like these things really are meant to be in a complex together, because, you know, as often happens with components of a complex, they can stabilize each other when you put them together. And I think that something like that may be happening here with smooth
And I think
NC
this
subunit.
cilia transition zone is very carefully
Yep.
regulated. What gets into the cilia, what goes up on these microtubules, and then, you know, it's very tightly regulated. And there are a lot of E3 ligases in there.
Yeah.
I don't actually think there are other regulatory subunits in the primary cilia.
Yeah.
Because this is R1, which is sequestered and bound to site with the, I think it's a lot of stuff going on.
So I forgot to repeat Susan's question. So we were talking about the transition zone of cilia, which is the part at the base of the cilium. I think that probably, I agree with Susan, that probably is the important location in the cilium where the key events are going on. That's probably where PKA is phosphorylating glee to keep the pathway off. Definitely, you know, we're continuing to kind of look at these things in primary cilia is going to be super important for sure. Yeah. Yes. Yeah, so the question is whether or not cancer cells change their ciliation status, ciliation profile. Yes, they definitely do. And so there's some examples of cancer cells that lose primary cilia as part of, you know, over the course of tumorigenesis. I think in some cases, it's a little bit challenging to interpret that because entry into the cell cycle and proliferation, you know, cells tend to resorb their cilia. But in the case of Hedgehog, though, it's really clear that the cilium is important for driving tumorigenesis, at least tumorigenesis that comes from mutations in PASH and smoothen. And so if you block formation of cilia, for example, then oncogenic mutations in PASH and smoothen they're very, very strongly reduced. So it does seem that it's going to play an important role in a cancer context too. Right. All right, any other questions or?
Thank you very much.
Thank you so much.